Hi, my name is Hannah Carson, and my senior seminar project was on the effects of temperature on decomposition rates, specifically studying decomposition in cold weather using fetal pigs. So, my thesis was that temperature, both warm and cold, have varying effects on decomposition rates in human remains. And so I examined temperature in greater detail along with other factors that are correlated to temperature. The purpose of the actual experiment really was to determine any effects of cool temperatures on decomposition rates using fresh frozen fetal pigs as models. So, why is this important? So, decomposition, specifically the process of it and the factors that affect it, are incredibly important to understand in the field of forensic science. One thing that's related to decomposition is the postmortem interval, or PMI, which is the amount of time between an individual's death and discovery of the remains. And in early stages of decomp, this is determined by forensic pathologists, while in later stages, it's determined by forensic anthropologists. And PMI estimation is commonly used in forensic investigations, for example, helping in victim and perpetrator identification. And specific instances of this have included being able to narrow the missing persons list to actually be able to use DNA or dental records or fingerprints to help identify the victims. And in terms of perpetrator identification, it helps in supporting or denying suspect alibis. So, what exactly is decomposition? So, human decomp is a complex biological process involving the two main stages of autolysis and putrefaction, where autolysis is when cell membranes and cell junctions are broken down by a series of chemical reactions. Putrefaction occurs after this, and that's when bodily tissues are broken down by microorganisms. There have been different studies have identified like various sets of decomposition stages, um, with one being fresh, bloat, active decay, advanced decay, and putrid dry remains, and that's based on the insect activity regarding to the remains. But there's other stages that are based on other things as well, and these stages are really used to observe the postmortem changes in the body and can really help in PMI estimation. So, there's a lot of methods in estimating PMI, but some of them are hard to use due to the different factors in decomposition. Specifically, a lot can be used in early stages of decomp, but as time since death increases, it really gets difficult to make an accurate estimation. Um, Early decomposition includes the fresh stage, in which characteristics of that include algor mortis, livor mortis, and rigor mortis. And those characteristics have really been found to estimate PMI with decent accuracy within the first few hours to first few days after death. But after the fresh stage, it really gets a lot more difficult because of higher variation. But some have found that insect activity, muscle degradation, and morphological changes of the remains have been used to sometimes estimate the PMI in these later stages, but it ultimately depends on the circumstances. But other fields in forensic science have been coming up with newer methods based on research in PMI estimation, including forensic botany, biochemical analysis of soil, the accumulated degree days or AGD method, and the degree of decomposition index or DDI method. So, these decomposition studies and other research surrounding decomposition have really been found to greatly help understand the process and factors affecting it. Previously, though, research was really limited due to the difficulty of obtaining human subjects, and so instead, these studies either consisted of retrospective case studies or was research involving animal surrogates such as pigs. And it really wasn't until the establishment of decomposition research facilities that this changed. With the first one being the Anthropological Research Facility at the University of Tennessee in Knoxville, which was established in 1980. Even as of 2018, there were only 9 in the world, with 7 being in the United States. Obviously, there's a lot more to do as um, the factors of decomposition really vary due to region to region. So, but yeah, These facilities are used to research human decomposition and the factors that affect it. There's a lot of factors that affect decomposition rates, which therefore also affects PMI estimation. The factors have really been classified into several categories, including environmental, individual, and cultural. And with environmental really being narrowed down further to biotic or abiotic factors, where biotic factors are caused by living organisms such as insect activity or animal scavenging, and abiotic factors are caused by non-living things such as temperature or other weather effects. Moving on to individual factors, those are include the person's health and body size. And lastly, cultural factors are when someone else does something to the body, such as an injury with trauma, embalming, or burial. And many have found that temperature is the most important factor affecting decomposition, um, along with being the most influential factor. And generally, it's well known that temperature determines the rates of chemical reactions. And so when applied to decomposition, you can see at warmer temperatures, enzymes will break down tissues a lot faster than in colder temperatures. And this goes the same uh, in the effect of bacterial and insect activity. At warmer temperatures, it's promoted, but in cold temperatures, that activity is suppressed. However, temperature is really affected by other factors as well, including being outside or inside, on land or in water, geographic region, sun or shaded areas, air movement, and altitude. So, beyond being affected by those other factors, temperature also controls the extent of other variables within decomposition, including humidity, bacterial and insect activity, and animal scavenging. Um, while temperature is affected by sunlight, sunlight versus shade really has its own effects on decomposition rates as well. Um, and in terms of insect activity, it's been found that it's directly correlated with temperature. So as temperatures increase, so does insect activity. And goes the same for as temperatures decrease, so does that insect activity. Another finding is that animal scavenging varies with temperature. One study by Zhang et al. Found that raccoons scavenged more when it was warmer in temperature than in cold temperatures. Moving on to the actual study. So for the experiment, two fresh frozen fetal pigs were used, both 14 inches, as to be as close to pig birth size as possible and to limit further variation. And the study took place in the small wooded area behind the Madonna University West Parking Lot A, which is between the Franciscan Center and the administration building. Two sites were chosen, with site one being a little bit more sunny area, and site two being a little bit more shaded area. And as you can see, the top picture is site one, and this bottom picture is site two. And again, as you can see, each site had a barrier of twine and these 4-foot tall garden stakes, and each site had a laminated sign with saying "Do not disturb" and my information and my mentor's information. And in order to prevent or limit any animal scavenging, dog crates were used and were secured to the ground. Data was collected 3 times between February 19th and 28th, specifically on the 19th, 22nd, and 24th, with pictures being taken additionally on the 21st and 28th. And data collected included temperature, weight, length, circumference or width, and pictures for visual observation. And additionally, weather data for the Detroit Metro Weather Station was recorded for each day from almanac.com, and they obtained their data from the National Climatic Data Center Global Surface Summary of Day. And as you can see, there was a variety of temperature data or weather data, including minimum temperature, mean temperature, maximum temperature, mean sea level pressure, mean dew point, total precipitation, snow depth, and various wind data as well. And here are the results for the weather data. Table 1 shows the temperature data in Celsius, and you can see that the temperature varied quite a bit over the course of the study, with a minimum at one point of -15 0.6 degrees Celsius. And of course, near the end of the study, the maximum temperature got all the way up to 15.6 um, degrees Celsius. Table 2 shows the pressure and dew point data. And of course, the pressure stayed mostly the same as normal, just varying slightly. But the mean dew point did vary a little bit over the course of the study. And table 3 shows the precipitation data. Not much precipitation, but as you can see, the snow depth started off at 13 inches and had melted all the way down to 1.2 by the end of the study. So you can see it melted quite a bit. And for table 4 shows the wind data, and of course that varied as typical. Moving on to the subject data, table 5 shows the data for pig 1. Table 6 showing the data for pig 2. And you can see there were some slight variations over the course of the study. Moving on to visual observation. So day 1 of the study on the 19th of February, there's snow on the ground and in both crates. And that was because the crates had actually been out a couple days before the pigs were put in there. And of course, both subjects were frozen solid. Day 3 was just visual observations, but animal tracks were seen at the entrance of the woods and around Site 1. And both pigs did appear visually less frozen. Day 4, it had snowed the night before, so there was snow covering both pigs in their crates. And they were both visibly and physically less frozen. And by day 6, the snow had begun to melt, and so some leaves could be seen through the crates. And upon arrival, actually, pig 2 was half out of its crate through a small gap in the front, with its left hind leg being eaten and its rear end being bitten. And the only thing remaining of its back leg, was a small bone found on the other side of the crate. And both pigs were thawed out with no odor, but there were some flies around pig 1. And then by day 10, which was the 28th, or on day 10, the snow had mostly melted with some mud in areas. And actually, both pigs were gone upon arrival on that day, with Site 1 crate being knocked over into its side with a swarm of flies around over the area like where the crate had originally been sitting upright. Site 2 being in place, but with having no insects visible, but there was no pigs present and no pig remains even there. So here's photos of pig 1. So you can see day 1, it's very visibly frozen solid. Day 4, a little bit less frozen, but still not thawed out. But by day 6, you can see it's very much thawed out. Here are the photos for pig 2. As you can see, like pig 1, day 1 was very much frozen solid. Day 4, again, a little bit less frozen, but still not thawed out. But by day 6, and you can see the bottom picture is what it looked like upon arrival. You can see down here, this is that little flap of skin is where is the leg that was eaten. Basically, the only thing that stopped it was its ribs. There's where it was eaten. And then there's the little bone that was all that was left. And then here's it laying out, and you can see there's where the bite was. And yeah, no left back leg. So the results were honestly inconclusive, and this was really due to the experiment ending a lot earlier than planned. It was planned to go until the 19th of March for an entire month, but it of course ended only after 10 days. But the data was starting to change as expected with the temperature, besides a couple strange increases in weight, circumference, and length. But the increase in weight is probably because of the pigs thawing out and potentially absorbing any extra fluid, which could also cause the increase in circumference in pig 2. But that increase in circumference could also be caused by the internal organs shifting around or potentially by bloating, but that's unlikely. The increase in length of pig 1 was definitely caused most likely by the thawing out and just a shift in like actually being able to measure it correctly. And then, of course, the decrease in length in pig 2 was caused by the anal scavenging. It was unexpected because the crates were secured to the ground. And while it was known that there was coyotes in the area, those crates were expected to keep them out. Moving even beyond that, it was surprising that once the pigs were gone, that there was nothing left as after day 6 there was a bone left. But if they were able to get the pigs out completely, it's most likely the case that whatever it was, whether it be coyotes or raccoons, took it back to its habitat. It was the primary assumption of it being a coyote due to the nature of the animal, and this is because it was actually found out that late March or late January, actually, sorry, through March, that's the coyote mating and breeding season. So they're a lot more active then. And they're also opportunistic hunters with a sharp sense of smell, and they're very adaptable. So, you know, they are more active, and so they're want to get whatever food they can get. Um, Moving on to some improvements to note for future research, definitely would suggest using some chicken wire or some like small graded fencing, metal fencing instead of dog crates, or at least to wrap the dog crates around in that to keep animals out. Another thing would be to potentially conduct the experiment outside of coyote mating season. While yes, the coyotes would still be present, they would be less active at least, and it would be interesting to see how that affected the data or animal scavenging at least. And then to using a motion activated outdoor camera, while this wouldn't, prevent animal scavenging, it would at least for sure explain or tell you when and what took the pigs. Also, it would allow more data to be shown for the animals that are around the area. And finally, using fresh pigs instead of fresh frozen ones, because some studies have shown that remains that are first frozen and then thaw out have different decomposition rates than fresh ones. So that would be interesting to see what the differences would be. And then in conclusion, from the previous studies, it's very clear that temperature is a very important factor in looking at decomposition and PMI estimations. From its effects on enzyme and bacterial activity to its effects on insect and animal scavenging, it's very clear that temperature is a very influential factor. And while the actual experimental results are inconclusive, this just shows that more research is needed for the region to be done. And finally, just some acknowledgments. I want to thank my mentor, Dr. Peckham, for his support and guidance during this time, and also to the university for financing this experiment. Finally, my references. Thank you.